Hi, I'm Climate Savage. I'm doing Biology A-Level, and today I'm going to be going over the structure of chloroplasts and how the structure enables them to carry out their functions. Now, I've done these videos before, and my computer went stupid, so no more videos. So I'm going to do them again, and which is good because that means I get to revise them more. So let's start off with the structure of chloroplasts. Now, if you have this book, is your biology? I've spoken about it loads of times before, but get it. It's really good, really helpful. So to start off with, that's ruined the illusion now, hasn't it? I just know this all straight from my head. I'm not using the book as like a guide or anything. Okay. Basically, to start off with, photosynthesis takes place within organelles called chloroplasts. Great, you should probably know that from year 7, year 8. And these vary in shape and size. Most are disc-shaped and between 2 to 10, I still don't know how to say it, little long. And they're each surrounded by an envelope, which is like a double membrane. There's an intermembrane space about 10 to 20 nanometers wide between the inner and outer membrane. And the outer membrane is permeable to many small ions, but the inner one is less permeable, and this has transport proteins embedded in it. It's folded into lamellae, which are like these really thin plates, and these are stacked up like piles of pennies. Each stack of lamellae is called a granum, plural grana. Sorry if you can hear my sisters, they're arguing. Okay, so inner membrane got transport proteins embedded into it, folded into lamellae, stacked up into granum. Lots of granum, grana. And between the grana are intergranal lamellae. lamellae. There are two distinct regions inside each chloroplast, the stroma and the grama. But both of these regions can be seen when chloroplasts are viewed under a light microscope. The stroma is matrix and it's filled with fluid, and the reactions of the light-independent stage of photosynthesis occur in here, in the stroma, because they've got all the enzymes that are needed in here. And within the stroma, there are starch granules and oil droplets. There's also DNA and prokaryote-type ribosomes. Moreover, the grana stacks of flattened membrane compartments called thylakoids. These are the sites of light absorption and ATP synthesis during the light-dependent stage of photosynthesis. And the thylakoids can only be seen using an electron microscope. So, how are chloroplasts adapted for their role? Well, the inner membrane, with its transport proteins, can control entry and exit of substances between the cytoplasm and the stroma inside the chloroplasts. Now, the many grana contain stacks of up to 100 thylakoid membranes, and because it's got loads and loads, this provides a large surface area for all of the photosynthetic pigments, electron carriers, and ATP synthase enzymes, all of which are involved in a light-dependent reaction. These photosynthetic pigments are arranged into special structures called photosystems, and these allow maximum absorption of light energy. Proteins embedded in the grana hold the photosystems in place. The fluid-filled stroma, remember it's in fluid-filled matrix. I really apologise to my sister. She's having like total meltdown. It's like after Christmas, so. The fluid-filled stroma contains the enzymes needed to catalyse the reactions of the light-independent stage of photosynthesis. The grana are surrounded by the stroma, so the products of the light-dependent reaction, which are needed for the light-independent reaction, can readily pass into the stroma. Chloroplasts can make some of the proteins they need for photosynthesis, finally, so genetic instructions in the chloroplast DNA and the chloroplast ribosomes to assemble the proteins. Great. I know I haven't really expanded on that. I've just sort of gone through bullet point. That's because there isn't that much to expand on. Like, you're not really taught that much else. You just need to know structure of the chloroplast. It's got this called lamellae, it's blah, and how they're adapted for their role. It's alright. It's not that bad. So, if you remember, I mentioned photosynthetic pigments. Well, these are substances that absorb certain wavelengths of light and reflect others. And they appear to us as the colour of the light wavelength that they are reflecting. And there are loads and loads of different pigments. and these act together to capture as much light energy as possible. These are in thylakoid membranes, and these are arranged in funnel-shaped structures called photosystems. And do you remember what they were held in place by? I said it in the how it's adapted. Yep, they're held in place by proteins. So we've talked about the pigments. So you should have heard of chlorophyll. Well, chlorophyll is a mixture of pigments. All have got a similar molecular structure consisting of a long phytol, which is like a hydrocarbon chain, and a porphyrin group. The porphyrin group is similar to heme, found in like hemoglobin. Remember what hemoglobin is? That's good. But it contains a magnesium atom instead of an iron atom. Now, light hitting the chlorophyll causes a pair of electrons associated with magnesium to become excited. And there are two forms of chlorophyll. There are two forms of chlorophyll A, P680 and P700, and both of these appear sort of a yellow-green colour. And each absorbs red light at a slightly different wavelength absorption peaks. It's like 680, P700, they're both very slightly different. And they're both found in the centre of photosystems, and they're known as the primary pigment reaction centre. P680 is found in photosystem 2, and its peak of absorption light is at a wavelength of 680 nm. P700 is found in photosystem 1, and its peak of absorption is light at a wavelength of, you've guessed it, 700 nm. Chlorophyll A also absorbs blue light of a wavelength around 450 nm. Chlorophyll B absorbs light of wavelengths around 500 and 640 nm, and it appears to be blue-green. I know that is a lot of different numbers in nm, but it's not that bad because obviously P60 absorbs P680 absorbs 680 nm, P700 absorbs light 700 nm. You just can't remember A and B, the difference between those really. There are also accessory pigments, and there are these such as carotenoids, and these reflect yellow and orange light and absorb blue light. And a good way to remember that is, it sounds like carotenoids, so you can remember that it reflects like yellow and orange light. They don't contain a porphyrin group and aren't directly involved in light-dependent reaction. They absorb light wavelengths that are not well absorbed by chlorophylls, and pass the energy associated with that light to chlorophyll at the base of the photosystem. Carotene, orange, see again, carotene, and xanthophyll, yellow. A good way to remember that, it's not that good, there aren't that many good ways to remember xanthophyll, but it's got a fill, it's got a Y in it at the end, YLL, so you remember that as yellow, and then it's got the two L's. That's a good way to remember it. And these are the main carotenoid pigments. So you must remember carotenoids reflect yellow and orange light. Remember, think of carotenoids. Carotene, think of really carotene. Oh, really important. Carrot. That's really the orange pigment. Xanthophyll ends in YLL, yellow. And these are the main carotenoid pigments. So that's basically how the structure of chloroplast enables them to carry out their functions. When answering questions, though, about light absorption, remember that pigments absorb a range of wavelengths. Refer to the wavelengths or the wavelength of peak absorption and not just the colour of the light. Want some questions? What range of wavelengths do you think that chlorophylls reflect? Do carotenoids absorb light of wavelength 500 nm? And you could also explain how the structure of the grana enables them to carry out their function. So, I hope you found that alright. It's not that bad. So, good luck with your biology revision and exams.